Right, so we are now being recorded. So welcome to this seventh video teleconference for BIOL 105 YA. We haven't done a video teleconference in almost a month, and that's because of the midterms week and also the other week that we allotted for sort of a break. So in this video teleconference, like the others, we are going to look back a review on the previous chapters that were covered. But I chose not to cover anymore the chapters that were covered in midterms. So for example, we were not able to do a video teleconference on photosynthesis, but since that's already part of the midterms, we will not include that in today's video teleconference. But tell me if you need a review on that. No, but anyway, that's already done. So in this video teleconference, our review will be on the extracellular matrix. That's the previous lesson from last week. Okay, so let's look at our calendar. Um, we are actually in sync with the number of video teleconferences that we're supposed to be holding. We are on our seventh. And because we skipped the previous week, we are now in sync with this calendar. So currently, in terms of lessons, you are supposed to be reading already chapter 8, maybe almost done with it. And I hope you've also watched the videos to help you in understanding some of the concepts. So, and again, as I mentioned, in this video teleconference, we'll review chapter 7. Okay. Right. So just like how we did it in the previous video teleconferences, I will be showing you pictures or figures again on the screen, and then we can start a discussion based on those figures. So before we do so, before we start, I'll just like to remind you that there's actually a quiz right now that's open, and the quiz is on chapter 7. So if you're attending right now, good for you. Because you are among the first in your section who will be reviewed on chapter 7, and you can take the quiz perhaps later on after this video teleconference, and you will be reviewed with some of the concepts. The quiz will be closing tomorrow, Saturday at 8 p.m. Okay, so you still have the chance also to take it tomorrow. Right, so let's start our review. So please, everyone is encouraged to actually speak up if they feel the need to share. Okay, and I also would appreciate it if you are able to add extra information that we might not be able to discuss. All right, so our topic is on interactions between cells and their environments. And I just like to point out that this particular topic is focused on multicellular organisms like plants and animals. Okay, because when we say the environment of the cell here, we are talking about the extracellular matrix, now the material that surrounds the cell in the tissue. So we're talking about animals and plants with tissues here. All right. So, but before we go to that ECM, let's go first to the one that's closest to the cell membrane of the cell. Okay. And that's what's being shown here. We have the cell membrane. But can anyone from the group um, tell us what these green branches are? Okay. So, or you may also include what this layer is and then what these red structures are. Anyone from the group? Okay, come on, guys. Let's get the discussion moving.
Uh, Ako, sir.
All right.
Um, the one colored blue and white is the lipid bilayer with the red color that is that has a part in the cell membrane is the integrins. And actually, I forgot what the green ones are called.
All right. Yeah, you are correct in those that you mentioned. Okay, we have the proteins. This could be the integrins and then the lipid bilayer. This one is attached. The function of this is for the attachment of cells or for recognition between cells. So, what biomolecule is that? Is it a protein? Is it a lipid? Is it a carbohydrate? Or is it a nucleic acid? Cell
Based
recognition.
based on what you said, sir, I think it's a carbohydrate.
All right. So these are carbohydrates. Now these are oligosaccharides. And many cells, like animal cells and even unicellular organisms, have this layer on top of their cell membrane. So, in fact, we call it, in a general term, that starts with letter G. Can anyone tell us what that term is for the carbohydrate layer?
Glycocalyx, sir.
All right, the glycocalyx. So in the video that I posted, I also showed their pictures no, of bacteria that are growing on an agar plate. No, I think you're familiar with bacteria, no, and then we culture them in the Petri plate. The colony of the bacterium, if you touch it with a material, it's like sticky, and that stickiness is actually due to the presence of the glycocalyx no, on top of the cell membrane of the bacteria. So the glycocalyx, because it's sticky, it also allows the bacteria to stick onto surfaces. For example, if you don't brush your teeth for like a day, no, when you feel your teeth with your fingers, it feels slimy and maybe a little bit sticky. No, and that's not just because of your saliva, but that's because of the bacteria that are thriving on your teeth surface already. And they are producing the glycocalyx, the carbohydrates. So, I think I believe we don't find this in plants because plants have cell walls, but animal cells and many bacteria have glycocalyxes covering their cell membranes. So, for multicellular organisms, particularly animals, we're talking about animals here, the cells are grouped together in tissues. No? So, we have here epithelial and connective tissue. So, can anyone from the group tell us the difference between the extracellular matrix in epithelial versus connective tissue? How does the ECM in epithelial tissue differ from that of the connective tissue?
Sir, I'm not
Yes.
sure, pero I think the extracellular matrix in the connective tissue directly bears mechanical stresses of tension and compression. I'm not sure
All right.
in epithelial tissue.
Okay. So the ECM in the connective tissue directly bears the tensional forces and compressional forces. Okay. You're correct. Okay. Where are the connective tissue in this illustration, no, in the drawing here? Um, If I may have the laser pointer again. So these light yellow substances between the cells, these are the ECM, no, including the collagen fibers that we see here, and also this dark yellow line here. Okay, so you are correct about the connective tissues ECM. What about the epithelial tissues ECM? What's the other name for the ECM of the epithelial tissue?
Is it the basal lamina, sir?
Yes, so you are correct. The basal lamina is also known as the basement membrane. Okay, so they're the same, basement membrane and basal lamina. So the purpose of the ECM in the epithelial tissue, as you see here, is what? These are the cells, and what are they? What is their relationship with the basal lamina? So these are the cells on top, and we have the basal lamina here. What's the connection?
Epithelial tissue adheres tightly to the ECM.
All right, so that's correct. For the ECM of the epithelial tissue, which is the basal lamina, the purpose is for the adhering of the cells on the epithelial tissue on that basal lamina. So basically, it's like a sheet of paper, and then you are sticking things on top of it. No? So, and then the things that you're sticking are the cells. So that's the basal lamina, and that can only be found in epithelial tissue. So that's an extracellular matrix. The story in the connective tissue is different. The ECM in the connective tissue is surrounding the cells in all direction. No, it's like the cement that is used in building a brick wall, and the bricks are the cells. So that's how the connective tissue may be perceived. So as your classmate mentioned a while ago, the ECM in the connective tissue directly bears the forces because they surround the cells. Okay? So I think that's clear enough for me as an explanation, but are there any other additional questions about the differences between epithelial tissue and connective tissue? What's an example of an epithelial tissue in our body?
Skin, sir.
Yes, the skin. And what about the connective tissue?
The dermis, sir.
All right. So the dermis of the skin, okay, is a connective tissue. Now, also adipose tissue is connective tissue, and blood is a connective tissue. So is bone and cartilage. All right. So let's go ahead of ourselves for a while here. I'd just like to ask you, what are these black things that we see here in the epithelial tissue? And what are these red lines? Can anyone tell us? Okay, I'll give you a choice. I go ahead.
Um, the black figures are desmosomes, and the red things connecting, the red things that span them are intermediate filaments made of keratin.
Yes, so you are correct. Okay, you are correct. Um, but the intermediate filaments are not really made just of keratin. Okay, so but you're correct that these are intermediate filaments. So these are desmosomes, and of course, this one at the bottom are hemidesmosomes. Okay, so hemidesmosomes here attaching to the basement membrane, and desmosomes attaching cell to another cell. Okay, so that's correct. And then we have the components, the molecules that are found in our extracellular matrix. We have 5 of them. Okay, so among the 5, only the integrin is attached directly to the cell membrane, as you can see here. The other 4 are strictly on the extracellular matrix. So we will try to talk about each one of these in the next slides. Okay, so I hope you memorize these and then their characteristics as well. Okay, that's very important. Okay, can anyone from the group tell us what's shown in this slide?
I think, sir, kanang nasa right kay collagen, and then ang
You
kanang
are correct.
nasa left kay kanang sa corneal stroma.
You are also correct. Okay, but both of them are actually collagen. Okay, so you are correct technically with your statements. Can anyone tell us further what's the difference between the two collagens? It's already said this one is found in the cornea. Okay, correct. So this one, where is this found? The one on the right.
So the one on the right is found on the basement membrane.
All right. So this is found in the basement membrane. So this is how the collagen in the basement membrane is arranged, and it's a special type of collagen. Type what? 1, 2, 3, 4, 5, 6, 7. Which
Type 4.
type 4 collagen on the basement membrane. While the one in the cornea, which are arranged orderly so that it appears transparent, is type 1 collagen. If I'm not mistaken. So you might want to review that. Okay, I believe it's type 1. So collagen is one of the extracellular matrix molecules. So can anyone in the group tell us the general characteristic of collagen? Any description at all about collagen?
Sir, it has a triple helix structure.
All right. So the quaternary structure is just 3 alpha helices that are intertwined with each other. So it's fibrous. And what important characteristic of collagen makes it um, invaluable as an ECM component? Unsa nga characteristic sa collagen? An example of this now is when you pinch your skin, when you let go, it will just spring back to its original shape thanks to collagen. So what characteristic of collagen is playing a role there?
High tensile strength, sir.
All right, so high resistance to tensile or pulling forces, no? Okay, so that's correct. Next, another component of the ECM. So this is, what is this? Anyone from the group?
Proteoglycans, sir.
All right, so proteoglycans. The name proteoglycan is actually a combination of two, di ba? Two names. Proteo because it has a protein. As you can see here, the core protein, no? In the branches here, the black line. And then glycan because the red lines that are attached to the protein are actually carbohydrates. So that's why it's called the proteoglycan. So can anyone from the group tell us the characteristic that's important in proteoglycans?
Sir, it has high compressional strength.
All right, so it can resist compressional forces, no? So it's like the opposite of the collagen characteristic. So if you put together a proteoglycans and collagen in the ECM will have the best of both worlds. The ECM will be strong in terms of compressional strength and also of tensional strength because you are combining the proteoglycans with the collagens, no? So in your skin, it's not just the collagen that's playing a role. There are also proteoglycans for high compressional strength. And that's because, if you remember the reason, what's the reason why proteoglycans are so strong compressionally? Parang trivia lang, if you can remember. Clue, it's because of the sulfates that we see here. What do they attract?
Water molecules, sir.
All right, so these sulfates attract water molecules, and in between the red rungs that you see here, there are water molecules there. So it's like putting styrofoam in between two layers of plywood. Diba? Styrofoam in itself is not strong, but if you pack it in between two layers of plywood, if you push the plywood down, the styrofoam will resist the compressional forces. So that's basically the same mechanism why proteoglycans are so strong compressionally. And then we have this molecule. So what is this molecule called? This illustration is actually not from your textbook, no? This is from another cell mall book. But you can compare it with the one from your book. I prefer this because it looks cleaner. So what is this molecule? Anyone?
Fibronectin, sir.
All right, so this is fibronectin. Can you describe the structure of fibronectin? Or
Um, it looks like duha ka chains, sir, nga naka-connect sa isa ka in one part, in one location.
yes. And the connection is a disulfide linkage. Okay? So if I may go back to the previous slide, no? Look at the fibronectin here. Where are they? They're the ones that are in purple. Look at the shape. It looks like a hair clip, di ba? Parang hair clip. It's like two strands attached together in one end. And look at this portion here. The fibronectin is attached to the integrin. Okay? And if we go back to the illustration here, there is an integrin binding site. No? Okay, that's where the integrin is attached. And that's because it contains the RGD sequence. Okay? RGD stands for the first letters symbol for the amino acids arginine, glycine, and aspartic acid, if I'm not mistaken. All right, so this is our fibronectin. Can anyone from the group tell us what the function of fibronectin is?
They serve as binding site. They have binding sites for binding with other components of the ECM, which connects them together.
Yes. So that is correct. So very obvious now in this illustration, fibronectin is able to bind to integrin and then to collagen, diba? And if you can remember in the video that I posted, they are also very important during embryonic development when the cells migrate from one place of the embryo to the other. The fibronectins serve as a pathway for the cells to move on to, diba? So please remember that one. And then we have this structure. What is this structure? So you may choose to describe the individual parts here, or you may choose to describe the whole thing. Anyone?
Sir, I think this is the structure of the basement membrane, and the ones in pink are the collagens, and the ones in green are laminin.
Yes, so that is correct. Now, so this is a drawing of the structure of the basement membrane. So you notice here, it looks like a mesh, no? Parang net. If we go back to that picture that I showed a while ago, this one, the type 4 collagen, these are the pink ones that you saw a while ago. So so itong mga pink ones, these are the type 4 collagen, as you mentioned. And the laminins are attached to the type 4 collagen as well to form the basement membrane. So the laminins have more or less similar function during embryonic development as this one, the fibronectin. Okay, they help in cell migration during embryonic development. All right, so for now, we have discussed those four main components, no, at least an overview of them because this is a review already. Now let's talk about this phenomenon. What's happening in this illustration? What tissue is this on top? Itong parang orange-yellow. What tissue is this? Epithelial or connective?
I think it's epithelial, sir.
All right, so epithelial tissue. And you can just see here the thick red line. This is the what?
The basement membrane.
All right, that's the ECM of the epithelial tissue. And of course, here at the bottom, you have the connective tissue. And you can see fibers, perhaps they are collagen. Okay, so what is happening here? What are these cells and what are they doing? Anyone? Are these normal cells? What's happening here? You might want to describe that. What are they doing?
Sir, they are cancer cells, which are, I guess, invading the connective tissue by using enzymes known as um, matrix metalloproteinases.
Right, so you are correct there. No? So you think of this, kasi ito, epithelial. So if we think of this as the skin, the surface of the skin, these could be skin cancer cells, no? That are on the skin, but they don't stay on the skin. They can actually penetrate the basement membrane of the skin, and they can go in through that, and they can spread to the other parts of the body, no, through metastasis. No, that's the process. And as you mentioned, they are able to do that because they produce an enzyme called matrix metalloproteinases. No, these are groups of enzymes that are able to destroy the extracellular matrix. Normally, cells use this to renovate. Not just like when you renovate your home, you break one wall of your home, you need to destroy the wall first before you can expand. Okay? So in the human body, the body does that as you grow through the use of matrix metalloproteinases. But cancer cells, like the one shown here, produce excess MMPs. Now, so they just break without any reason um, the ECMs of the tissues that you have, so they can spread in your body and they can invade new areas in your body. So that's the reason why skin cancer can spread to the lungs or to the liver or to the kidney, but they've actually originated from the skin. Okay, so question: what cofactors help MMPs do their work? Ano ang mga cofactor ng matrix metalloproteinases? Because enzymes need cofactors in order to work. Clue, they are inorganic ions. Clue, one of them has a positive 2 charge, the other has a plus 1 charge. And they are metals. O sige, so what are they? What are the cofactors of MMPs?
Calcium and zinc.
Okay, calcium ions and zinc ions. Now, among many others, but they are the most common ones. So, with the help of calcium and zinc ions, the MMP can do its work. And the cancer cell can also invade or eat through the extracellular matrix. All right. So, and then there is this part of the ECM that's attached to the cell membrane, as we see here. So here on the left, they are inactive. They are dimers composed of alpha and beta subunits. And when they detect a signal from the outside or from inside the cell, they become activated like this. What are these? The green structures.
Integrins, sir.
All right, so these are integrins. Okay, so the name integrin could have many origins. So one is that they are integral proteins. So look at this, they pass through the cell membrane, the bilayer. Number two is they integrate signals from the outside of the cell, no, like this collagen. This is actually a signal that the cell can detect, and then the integrin will activate, or the signal can be from the inside, like this talin molecule attaching to the beta subunit of the integrin, also activating the integrin. So integrin, because they're integral proteins and they integrate messages from the outside of the cell to the inside of the cell and vice versa. And they also serve as the feet, no, of the cell. If you have held a starfish before, no, you notice those tube feet at the bottom of the starfish. I imagine integrins like those. No, they're like the small feet of the cell, and you can actually see the cell here doing its work. So can anyone from the group tell us what's happening to the cell here? So I think this is step 1, step 2, and then step 3, and then step 4. What is happening to the cell in these illustrations?
The cell is using some sort of attachments to attach itself to the flat surface.
Yes, okay, so this is a glass surface, no, maybe in a petri dish, and the cell, the live cell is placed there, so the cell is attaching itself using what? Using this, the integrin, okay? So the cell is attaching itself onto the surface using integrins. And it's like when I let you ride a roller coaster, and then the roller coaster is about to do a vertical turn, you hold on to the railing really tightly so that you won't fall. So the same thing is what's happening with the cell here, so that the cell will not roll when it's handled in a Petri plate. It actually secures itself. It holds onto the surface of the glass using integrins like this. And how do we call this type of attachment in cells? There's a term for this.
Focal adhesion, sir.
All right, so this is focal adhesion. And we see it here now in this colorized micrograph of the cell. You can see the integrins, the yellow dots, and they are actually attached onto the actins, the green filaments. So in my own description, the integrin is like the hands holding onto the surface, and the actin is like the arms that are attached to the hands. No, so mangusog yun ang cell in order for it to stay attached to the surface. So I think you can all relate to that. No, you can relate to the cell. When you are, this happens to the cell when the cell is placed in a non-native environment. No, when you remove it from its normal environment and you put it in another environment, so mahadlok ang cell because it's quite alone, so it will hold on to things really tightly using focal adhesions. So here's another view of that. You can see here the glass surface, and you can see the integrins like the tube feet of a starfish, and they are attached to actin filaments, the blue ones here. So that's focal adhesion. But when the cell is in its normal environment, like when it's on the basal lamina, what type of adhesion does the cell use? We already mentioned this a while ago.
Hemidesmosomes.
All right, so it's shown here now. This is the basal lamina of the epithelial tissue. It's the hemidesmosome that the cell uses to attach onto that. And what is this filament? This is intermediate filament no, ang naka-attach instead of actin. Okay, so what's the difference? Actin is the one found in your muscle. No, actin can exert force, can exert effort, but intermediate filaments are like para siyang kabilya, no, the skeleton of a building, the iron skeleton of a building. It doesn't exert force, but it's strong enough to withstand force. So the cell uses the hemodesmosome when it is in the normal environment because it doesn't have to hold on too tightly. It just needs to stay put. So okay na ang hemidesmosome. Right. And you notice here what is being attached to the hemidesmosome. You have the integrins here, the green, attached to the laminin in the basement membrane. And you have here in blue the type 4 collagen. Type 4 collagen. Okay, so that's for the hemidesmosome. Now, let's talk about cell junctions. Okay, we have here 1, 2, 3, 4 junctions. Anyone from the group pick a junction and tell us about it and its function. Okay, just any junction lang guys and you say something about it. Hurry up because time is moving really fast.
Well, sir, gap junction, sir.
All right, we have the gap junctions.
From what I remember, the gap junctions are the ones that connect uh, the cytoplasm of two cells together, and that's how they like pass through ions, as also seen from the figure.
That is correct. No, so they can the gap junctions serve as communication channels between two adjacent cells. And you termed a while ago they serve as connections. Therefore, the protein that make up these each of these channel are known as connexins, and each of the channel is known as a connexon. Okay, so that is correct. Next junction, please. Anyone?
Um, tight junctions prevent the tight junctions seal the membranes of two adjacent cells together, and which prevents the movement of material in between them. And they are composed of claudins, I think.
Okay, so everything you said is correct, no? So the name of the protein that makes up the tight junction, what I remember is a cloudy day. The clouds prevent the sunlight from passing through. So I remember cloudine because they prevent materials from passing through in between the cells as we see here. So everything was correct there. Next junction, please. We have two more junctions. I'll give you a head start. These two junctions, adherens and desmosome, have the same function. It's for the adherence no, between two cells, the adherence between two cells. So my question now is this, how are they different? They have the same function, but how are they different? Very obvious already from the illustration, guys. You can just see the labels.
Adherence junctions use actin filaments while desmosomes use intermediate filaments.
All right, so that's correct. No? So the desmosome, similar to the hemidesmosome, is attached to intermediate filaments inside the cell, while the adherence junction is attached to the actin filaments. As I mentioned before, the actin filaments are more stretchy and they exert force, while the intermediate filaments are just there, but they are strong. They're like the foundations of a building. So notice also the positioning adherence junction are towards the top while desmosomes are towards the bottom. All right. So what about in this illustration? No? So still on cell and cell adhesion. This shows us the formation of the neural tube in an embryo. No? So here we have a layer of cells that are all the same. Here, somehow they become different. There are now two groups. And then the other group separates from the first group and forms a new group, the neural tube. So, anyone tell us the background why they are now separating from each other? What's in the blue cell that's not in the orange cell? Why do they need to separate?
The cells in the blue part of the membrane use a different kind of adherence, um, which means that they no longer recognize themselves as part of the orange part and dissociate to form their own membrane.
All right, so cadherins are cell-to-cell attachment molecules, no? Cell adhesion molecules that allow one cell to recognize a similar cell to itself. And similar cells will attach together using the cadherins. So somehow along the way, the blue cells that we see here have expressed a different kind of cadherin as compared to the surrounding orange cells. And so the cells here that are in contact with the orange cells recognize themselves as different already. And so they separate. So what you said is correct. No, it's because of the different expressions of the cadherin molecules. So remember that between the same cells, they are together because they recognize each other because of cadherin. Okay. And last but not the least, we have this junction in plant cells only. How do we call this? You can see here the cell wall, and you can just see here these gray lines, regions. They allow communication between one cell to the adjacent cell in plant cells. So how do we call this channel?
Plasmodesmata, sir.
All right, so plasmodesmata. What is their equivalent in animal cells?
I think gap junction, sir, or
You are correct. So gap junctions no, in animal cells. So sa plant cell, because there's a cell wall, it's a special type of gap junction and we call it the plasmodesma, singular, plasmodesmata, plural. And then a special type of extracellular matrix is the plant cell wall. No? So the plant cell wall is made up of this cellulose microfibril first, no, made by the cellulose synthase that are on the cell membrane. And the orientation of the microfibril is dependent on the microtubule underneath the cell membrane. And there are also other components like the hemicellulose that we see here, and then the pectins, and then the glycoproteins. So last question, okay, because this is the last slide. Can anyone describe to us how the cell wall of plants is made? How is it made? I already mentioned a portion about cellulose synthase. What's happening here? What is this illustration at the bottom right showing?
The cells in the right show, in the bottom right, show how materials in the extracellular matrix of uh, plant cells are synthesized inside the cell and are carried via Golgi complexes and on some others towards the plasma membrane.
Yeah, okay, that's correct. So if the cellulose microfibril, the big brown fibers here are made by the cellulose synthase, the other stuff of the cell wall, like the hemicellulose, glycoprotein, and pectin, are made inside the cell. And they are carried onto the surface via the Golgi body and then the vesicles to be deposited on together with the cellulose microfibril. So you think of this, guys, as like this. First, no, in Bisaya pa, ang kabilya sa building ang ibutang, di ba? The foundation that's made of metal, the wires ba yun? The poles, the metallic poles, the kabilya. I don't know the English of that. Okay? So, after the kabilya is done, what do? You then put the hollow blocks, you then put the cement. So, that's how the cell wall is made. First, the kabilya or the cellulose microfibril. And once that is in place, the other parts, the hemicellulose, the glycoprotein, and then the pectin are then added from the inside of the cell. Okay, so that's how the cell wall is made. So thank you for listening, no, and interacting with me. Many of the things that we are discussing here will come out in the quiz. So if you haven't taken the quiz yet, um, I hope this one helped in the review for the quiz. So the quiz will close tomorrow, Saturday at 8 p.m. The group discussion for chapter 7 was already posted. Now I hope you have accomplished that one, the one in the table. A new group discussion is still to be posted, and I will just tell you if it's ready. But as of now, you don't need to worry about that one. And one last thing, your grades are not yet done. I am still very busy checking stuff because I have your laboratory. I have the laboratory of many sections as well. I have genetics, and then I have this, and I have a lot of things to do still. So don't worry about that. It's gonna be posted soon. That's my problem if it's late. But I will tell you once it's posted. So please be patient with me on your grades for the midterm. Okay, so other than that, we are done. And are there any questions or concerns? Time check 9 a.m. So we are on time. Questions, concerns? Silence means there are none. Okay. So I'd just like to say this that thank you for joining this video teleconference, and it's really important not to have someone to be present in a video teleconference, even if it's not all of your classmates. Why? Because if no one will come, then I will not be able to do this discussion, and I will not have a recording of this, and therefore your other classmates will also not benefit, because there's no recording, there's no new discussion. And I really want these video teleconferences to be reviews, so it's very important for them to really happen. So thank you for making it happen, for attending this video teleconference. So other than that, because you don't have any questions now, you are now free to go to your other classes, so you are now dismissed.
Thank you, sir.
Thank you,
Thank you,
You're
sir.
sir.
welcome.